it's great to be here at the OIS 2024. So we're going to kind of jump right into it. So the idea here began with, what's the best way to learn how to do surgery? What's the best way to develop devices? What's the best way to probe the ophthalmological vision system for development and research, right? So our answer is near real surgical specimens, these kind of tissues that are like everything. No one's ever been able to tell in our blinded test if it's a live human or a cadaver or one of our NRSS models until they touch them because of the temperature, they're cold. So currently what's available is a myriad of things that range from fixed like plastic, rubbers, silicone models, and then kind of augmented reality and artificial reality. And so these models do usually very well at sometimes doing one part of a procedure, one specific part component. However, where they fail is when we know this from our Surg Tech AI modeling of surgeon behaviors, that the surgical proficiency performance builds on the previous step. We all know this. And so really it's with NRSS that you can put together the whole surgery just like you would for anything else. So the need here is, and this is a proof of concept, we don't use any tissue fixatives. That's the trochlear nerve going through the trochlea. In any formaldehyde-based model, this is impossible to see because you lose the trochlear. The formaldehyde would basically cause it to break. And so here show that there's no smell. These things can be in your fridge for about 10 months. You can do an infinite number of intravitreal injections. You get about 4 conj procedures, like 4 tubes, 4 trabs type thing. You get 1 lens procedure, 1 lens rescue procedure, 1 retina procedure, multiple strabismus per depending on what you're looking at. And so really what we're trying to achieve and get to is kind of mimic the structural and functional properties of the human eye. So this is here we're measuring, you can measure and you can start having different processes to look at what's the optimal suture configuration for force vectors when you're doing a corneal transplant. So you get to answer questions that are meaningful and impactful that we don't typically have the opportunity to address in typical clinical programs. So what do we get? We get enhanced training outcomes, we get high fidelity simulations, and we get durability and reusability across. And so we have validated models in oculoplastic, strabismus, cornea, glaucoma, retina, sclerofixation, and cataract. The one on the top left here is from our Thea project with DARPA, the transplant of human eye allograft, a full eye. So that's our nerve model there that we used. And so ultimately, this is what we got to get to. I think the way we're developing devices, especially currently, is how we're developing drugs. But they're not like drugs, right? If you could, here you're seeing our 3-camera view. There's a camera in the optic nerve, a camera on the outside, and there's an OCT camera running. And so for things like that are going into the eye, that you can now measure the force vectors. How big is that wound? How many sutures? Should you put a suture on the end? Should you do a mattress? Should you do a cross stitch? In our retina model, you can get down to all these. And I think this is how we kind of bring it out into, rather than having a long clinical development profile where, hey, now you finally do this thing, you can beta test your device that doesn't need humans. That is to say, no human is hurt with any of this testing. So as soon as your model's ready to be tested again, you take it to the NRSS, you test, you repeat it, you go back to the lab. And then when it's actually ready for a human, bring in a human. Because a lot of that, especially with devices, early phase testing doesn't require humans because you end up getting actually confounders because now you have some treatment effect in there and how much was that proficiency of the surgeon learning a new technique. When we look at the competitive landscape, thinking about plastic silicone models, human cadavers, and then advanced tech like VR and AR simulators, we know this, that NRSS are the only ones that give all of these, where it actually gives you realistic tissue texture and rigidity, reproducibility, validity, and then when you actually model and test these surgeons when they're learning new techniques, whether it's a suprachoroidal delivery of a treatment, this is usually how we find they learn the best. This is a protected intellectual property. And so we know there's a big global market because there's nothing else like NRSS that exists, right? We have big medical education market. We have our ophthalmic market, but also have a very interesting non-ophthalmic market that you can penetrate like with derm, definitely with derm and plastic surgery and cosmetics. Those are going to be the big ones on the non-ophthalmic side. And our 5-year model, because of the lack of competition in the market that could develop and gain from a beta testing program, we see this is a positive figures because we have these kind of competitive advantages. We have a first mover advantage. We have a very scalable business model. We can make as many of these as we need. We have a protected IP. We have an experienced management team that on 5 continents that deploys this and it's proprietary technology. How we engage with them is a little bit different. So this is not something you order on the website and the head arrives at your place. So we basically sit with you and say, hey, what are we trying to achieve here? Because we can create a validated model specifically for your device or for your problem. And I think this is when it really gets into what are the possibilities where that we can get to. We took the 5 devices that were approved by the FDA, the last 5, the camera inlay, PreserFlow, the LensX, the Illuvian implant, and Roche's Susvimo. So if we went into the early part of it, where there's research and development around the device, that first part of the development program, this is the kind of cost savings that we can get. We're looking at 20, 30, 40% of savings at a time when you're trying to make that decision. It's like, should I kill this thing, or does this thing have a life beyond this trial? Or so this is where we find this arbitrage, where we now can help you bring something to market faster. We know way more about it in terms of what it's doing to the eye, and the safety is, you see that paramount effect right away. Currently, we're in a $7 million seed round to help for expansion on 5 continents. And then there's one, and yeah, thank you. There was one other video here, but I guess it got dropped. So one of the things that you could do is, so we have one of the videos is something that we took. Basically, we can take these models, and for example, one of them we froze one with liquid nitrogen at -90. We then basically use a temperature gradient to bring it back to normal temperature. You put a drug in a drug-eluting silicone wafer, you put it on there. That's a very nice Q-sub-regulatory approach where you can have a drug in there. The device never went to the patient, gets thrown out. So these are the kind of things that you could develop and that we bring with the NRSS. So thanks so much for your attention, and look forward to chatting more about them. Thank you.